A very good evening and welcome to one and all present here for DENTOPHARM International Webinar Series on topic Cold Sensing by the Heat Shock Proteins. I'm Dr. Ramya, part of Dental Education Unit and Senior Lecturer at Sri Balaji Dental College and Hospital. We'll be highlighting you about this program today. I would like to welcome our guest speaker, Dr. Raghawan from Harvard Medical School, and I also welcome Chairperson Dr. Srinisha Ma'am, Registrar Dr. Bhuminathan Sir, Principal Dr. Jimsy Sir, and Dr. Sumati Jones Ma'am, Head of the Department of Pharmacology at Sri Balaji Dental College and Hospital. Over to Sumati Ma'am to share few words on today's webinar.
Welcome to DENTOPHARM webinar series. We have Dr. Raghavan with us. Dr. Raghavan sir will be explaining about cold sensing by heat shock proteins. We all know that heat shock proteins are involved in folding and unfolding of proteins during stressful conditions, and it plays a key role in cardiovascular diseases, immunity, allergic reactions, and so on. Sir will be the right person to explain about it in detail. Over to Ramya again.
Thank you, ma'am. Now, I warmly welcome our guest speaker, Dr. Rahman, who is postdoctoral fellow in the Division of Beth Israel Deaconess Medical Center at Harvard Medical School, Boston. He obtained his Bachelor of Science in Chemistry and also Masters of Science in Molecular and Human Genetics at Institute of Science in Banaras Hindu University in Chennai, in India. He pursued his PhD at Center for Cellular and Molecular Biology in India. He has worked in various research areas to fellowships, and he also received Professor S.R.V. Rao Best Paper Presentation Award at 43rd All India Cell Biology Conference in 2019. Recently, Dr. Raghavan was nominated as nominee for Indian National Science Academy Medal for Young Scientist 2021. After working in various levels of research, Dr. Raghavan moved to the United States to pursue postdoctoral fellow at Harvard Medical School. His work has provided insight into how the heat shock protein sense the cold temperature and regulates inflammatory response in a temperature-dependent manner, investigating the pathophysiology, cold-inducing aggravation of symptoms of arthritis and related disorders. We welcome you, sir.
Thank you. Thank you very much. And I'm really overwhelmed with this introduction. And thank you for this opportunity to present my research work and also interact with students, especially the clinicians, the future clinicians. And I will be very happy to start right away. I have two agendas here. One is, of course, I will try to take you through my own research career till now, which is approximately 11, 10 or 11 years, and in which we have discovered a few interesting things. Apart from that, I added one topic to my presentation, that is relevance and rewards of research. Only reason for that is in India, I felt that there was no doctors in our research laboratories. Maybe 1 out of 1,000 people who do research work in laboratories are also MBBS or dental students or some things like that. None of them are clinicians. When I came here at Harvard Medical School, there is a sharp contrast to that population. Almost half of the people who do research in laboratories are also clinicians. So they work like triple shift in a day, 2 shifts in the hospitals and 1 shift in the research lab. And only half of the population is pure researcher, like who is not a clinician. Now, this makes a lot of difference in our health sector, and that is why I will also try to attract you while giving my presentation towards research. Because no way me, who is not a clinician, who did not get a training like MBBS or dental science, I cannot go to clinics now, but you can come to research. So I will also try to attract you towards research during this presentation. I don't know how it is going to be perceived, but okay, I will try. Uh, so let's start with the science part. If for any reason you do not understand the experiment or the data in my slides, just ignore it. Don't worry about it. You can continue to just listen what I am saying, and I will try to give the essence of the experiments that is performed in scientific laboratories, research laboratories. So there are some things which are very common that we both of us know, the researchers as well as the clinicians, that is, let's say this is a infection when a bacteria or any other pathogen invades an immune cell like macrophage that phagocytoses that bacteria or the pathogen. After the phagocytosis has happened and the bacteria or the pathogen is inside the cell has to decide if it can clear off the bacteria, if it can kill and digest the bacteria. And uh, if that is the case, then it will undergo inflammation. That is, it will digest the bacteria and release cytokines like interleukins, IL-6, IL-21, IL-1 beta, IL-17. Many of these IL-6 and all we are now everybody is acquainted with during the COVID times. So these interleukins are cytokines that will attract other cell types, and all that cell types of the immune system will try to uh, uh, phagocytose and digest and clear off the bacterial infection. Now, this happens only if the cell thinks that the bacterial load, the infection, is within its control. It is not too much. If it is too much and the cell is overwhelmed with the bacterial population, then it will undergo apoptosis. So that is cell death. So it will uh, die in a very regulated process, and along with it, all the bacteria inside the cell will die. Now, this is a altruistic behavior, a sort of it is dying, it is undergoing apoptosis to protect other cell types, all other cells of the body. So this decision whether to undergo for a survival and inflammatory pathway or to undergo for apoptotic, that is cell death pathway. So this is a decision between life and death that any immune cell has to take after infection. Now, there are several immune receptors that can detect bacteria in our body, and there are n number of them. Many more are being discovered that these receptors inside the cell are the sensors to a pathogen or the bacteria. Now, I just mentioned that decision of life and death is taken by every immune cell immediately after the infection. When I started my research career, this was the question that how this decision is being taken. Is there a counter inside a cell that counts the number of bacteria? Okay, now it is above 100, let's die. It is still 99, let's try to survive. Is it that? Or there is a particular biological phenomena that regulates this process, that, okay, this is how it will be decided whether we are continuing to survive or we should go for apoptosis. So this was the research question that I started with during my research career. Now you can see there is a protein NLRC4. This NLRC4 is a receptor. That is, it is this is the protein which detects a bacteria or any other pathogen. And this protein can trigger inflammatory pathway, that is caspase 1 activation. This is the survival pathway on the left side, and on the right side this sub1 ubiquitination and caspase 8, that is the cell death pathway, the apoptotic pathway. So after sensing of the bacteria, NLRC4 is the protein which will be involved in the decision of life and inflammation, or death or apoptosis. Now, when I joined the research, this was amazing question. You are going to discover the switch between life and death. And we did a lot of work in this field. Um, mm, we uh, we identified some of the factors, some of the proteins that are part of the team that take this apoptosis pathway. uh, that will come in the later slide. And here, if you are interested, I will just show you how the inflammation pathway works. So this is the cell. When the bacteria is phagocytosed, NLRC4 detects it, and then the NLRC4 gets activated. Usually in a cell, whenever protein interacts with another protein, its shape changes in a certain manner. And once NLRC4 is like this, it is very similar to like this. This is a cartoon which I have prepared, but inside the cell also, this protein looks like in a circular, closed circular loop manner, like a ring. And once the bacterial fragments are detected, this ring opens and this becomes in a question mark kind of shape. And then it interacts with other proteins, one ASC and procaspase 1. And then this is an inflammasome. This is also called death of wheel in the research field. And inside the cell, you can actually see these kind of structures in electron micrographs, very symmetric, very circular, like a spoke wheel structure. And then afterwards it activates certain enzymes, procaspase 1 and pro IL-1 beta, and this is the IL-1 beta which is getting released into the outside the cell. And this is the inflammatory pathway. I have painted it green because it is the survival pathway. There will be conditions when NLRC4 will be mutated. There will be certain genetic mutations, and all the clinicians will know about the ill effects of genetic mutations. Now, what happens if there is a genetic mutation? If there is a genetic mutation that, even in absence of a bacteria, makes NLRC4 in an activated stage, like in the question mark stage, it will continue to form these inflammasomes. It will continue to activate caspase 1, and it will continue to do inflammation. Now, inflammation is good when there is a bacteria or any other pathogen, and then you have to mount an immune response. Inflammation is not good when there is nothing to defend, and you are just killing your body with more and more and more and inflammation. So, in order to identify what NLRC4 does to make the decision between life and death, that was the question to start with. We used certain genetic tools, certain genetic mutations of NLRC4 to see the effect. So let's say it is like it, there is a toy on just lying on the road, and you don't know whether it works or not. So what you do is that whether the toy is functional or not, or what kind of running this toy will do, whether it will just walk slowly, or it will be a fast toy car, or it will not run at all. It is a dead toy car. So if you have to test it, you have just found it on the road, you don't know anything about it, who it belongs to, when it was bought, where it was manufactured, you know nothing. But you just know that it is a toy car. It may be a toy car which will run very slow, which may run very fast, or which may not run at all. What you do is you put a battery, you switch it on, and just leave it on the road, and then just watch what happens. If it is running fast, then you conclude that, okay, this is a fast toy car. It is a slow toy car or something like that. So here also, we don't know what NLRC4 will do once it gets activated. So what we do is we put a mutation in it, and mutation, and then we just look at what it did. whether it get hyperactivated, whether it did not get hyperactivated, whether it is there is no difference in the phenotype. And so we did not just make any mutation. We found some mutations which were reported by clinicians, of course, in the hospitals. And these mutations were supposed to cause certain diseases. So S171F or T, these are the amino acids. And this amino acid is at once a serine amino acid converted to F, tyrosine to alanine at 177, tyrosine to threonine to serine at 337. So all these mutations are known, were told to us by clinicians. Well, that this mutation happens and this protein becomes hyperactive, it undergoes a lot of inflammation. All these mutations except this H443P actually kill the infants. By the end of first or second year, the infant gets killed. We took this H443P mutation that allows the people to survive. There is no effect on the lifespan, but there is unique thing. Whenever these individuals face cold weather, so as long as you are at 37 or above in a warm temperature, you are absolutely normal as your neighbor. But as soon as you are exposed to cold weather, chilled wind, cold water, you start showing symptoms of hyperinflammation. So this was very interesting, that there is something that keeps you okay while you are at 37 or normal temperature. But when you go to cold temperature, something happens and you just fell sick. And then we had certain objectives. I won't go into those objectives. Right now, it is not of concern. Um, these are some images where you can see a cell inside your body. Now, this image, we just collect cells from the human body and put it under the microscope, and you express those mutant proteins inside the cell, and then look at the effect of it. That what happens to the cell, whether the cell is dying, whether cell is having no problem at all with these after expression of these mutant proteins. Uh, just look at what happens. And we observed that the cells begin to die. If you express the H443P mutant, the cells, this is a dying cell. You see, can if you can see, there are fragments going off from the cell. So this is the apoptotic cell here. And then you can also monitor certain protein levels. So whenever the cell starts to die, certain proteins will increase inside the cell. So now we know that the caspase 8 gets activated or caspase 3 gets activated inside the cell. So when the cell is not dying, there is no caspase 3, but this protein gets increased when the cell is dying. So these are some of the experiments which we perform in the lab. So you put a protein inside a cell and then look for the effect. And this is very simple process. You look for the effect under the microscope, or you can take all the protein of the cell, run it into a gel, and then look for the protein levels. And these two things will tell you if the protein is higher, the cell is dying. If you can see the cell dying directly inside the microscope, that is the best way, that is the best confirmation. So we just did this test and we found that, all right, protein H443P kills my cells. Similarly, we did other type of experiments. In the previous experiment, we overexpressed a mutant protein H443P inside the cell and we looked for the effect. In the next set of experiments, we did is that we reduced the amount of protein inside the cell. Now we have certain tools to do that. You can actually remove a protein from the cell by using siRNAs. So these RNAs go and bind to the mRNA. All of you know that the gene transcribes mRNA, and then mRNA gets translated into the protein. So if you remove the mRNA from the cells, there will be no protein formed. So if I want to remove certain protein, let's say SUG1 is a protein, and if I want to remove that protein from the cell, you remove the mRNA from the cell. If you want to remove the mRNA from the cell, just add something to the cells that goes and binds to mRNA and degrades it. So that is what we did. We did shRNA and siRNA, and these are very simple techniques, very straightforward techniques if you want to see effect of a presence of a protein or absence of a protein. So here we we applied this method, that we removed the mRNA of SOG1 from the cells, and we looked at the effect, and we found that H443P mutation is not able to induce any more cell death. Now, we added HP mutant, it kills the cells. You add HP mutant, but you remove SUG1, the cell doesn't die, which means HP mutant requires SUG1 to kill the cells. Likewise, we also tested what are the other factors. Okay, the SUG1, there is some other biological phenomena, that is ubiquitination, that is required for uh, cell death. We felt that okay, apart from SUG1, ubiquitination is also required for cell death. Then, is there anything else that is required for the process, that is required for the cell death? From the literature, many people have done this research that FAD is another molecule. Like SUG1 ubiquitination, the FAD is another molecule. Whenever there is a cell death happening, this molecule is likely to get involved. It is like it's kind of the Yamraj of for the cells. It is always there whenever you want. So. We also tested whether FAD is also involved in this process, the cell death caused by NLRC4 H443P mutant. And we did a lot of experiments. Every time what we do is we overexpress this mutant, we overexpress this mutant, we overexpress SUG1 plus NLRC4 inside the cell. So all these 5 factors, they cause cell death. But if you remove FAD from the system, FAD from the cells, then they are not able to cause cell death. And we came to this conclusion that no matter what, NLRC4 HP mutant or NLRC4 ubiquitin mutant or NLRC4 along with SUG1 or another form of NLRC4, all these forms of NLRC4, they lose their capacity to cause apoptosis if there is no FAD. So by this time, we have formulated a line or a series of experiments and formulated a model, formulated a proposal that this is what happens inside a cell when apoptosis happens. And our model was like this, that NLRC4 interacts with SOG1, or if there is a mutation, then NLRC4H443P mutation that interacts better with SUG1, it undergoes ubiquitination, and then it engages with FAD and caspase 8, and then there is activation of caspase 8, and then there is apoptosis. Now, this was the process of research. Uh, we used, we put some proteins inside the cell, we removed some protein from the cell, we analyzed the cells under the microscope, we looked for which protein is going up and which protein is going down using different techniques. We also looked at which 2 proteins are interacting with each other. There are very simple techniques like immunoprecipitation or things like that. And we now know what happens inside the cell when a cell decides to die. That decision is made by NLRC4. At the end of this, yeah. So we have At the end of this, we have defined this path, the right hand side path of the apoptosis, and everybody else in the world, like there are hundreds of lab that they are working on the left side, the inflammation path. From India, working at CCMB, we defined this path, which is exclusive. Nobody else has shown, apart from our lab in CCMB, about the cell death pathway. So now we have added to the information that NLRC4 can induce the survival inflammation pathway as well as the apoptotic pathway through SUG1, ubiquitination, caspase-8, and FAD. Now, this was the process of research which gave us satisfaction, and now the companies like pharmaceutical companies will look at this diagram and look for, well, what can we inhibit? Can we inhibit ubiquitination? Can we inhibit interaction of FAD with an HP mutant? Can we inhibit caspase-8? So they will try to find out inhibitors. If you can inhibit any of the process in this whole length of any of these arrows, you can inhibit cell death. And then you can have a drug for a disease where there is a lot of cell death. So if a disease that is caused by excessive cell death, you can have a drug by inhibiting any of these arrows. And that is what the pharmaceutical companies do. So our research has the potential, I should say, to give rise to a new drug where excessive cell death will be a problem. Okay. Now, I'm not saying that this diagram is going to lead to a drug development. I'm just giving you an example of how the drug which clinicians prescribe in the hospitals to their patients is developed and what happens in the background. Okay, the next part of my talk will be maybe 15 minutes or so, and here I will try to explain you that how you can, let's say, find more satisfaction while doing research, or what is very critical to our research process, the process of our research. The process of research is like, the basic and the most critical part is observation. You know, all of you have written an essay on, let's say, a cow in your childhood. That must be your first standard, class 1 or class 2. And when you write your first essay on a cow, what do you write? We write a cow has two eyes, a cow has one tail, a cow has four legs, and that's the simplest, that is the simplest way of writing an essay. That is also the simplest way to observe anything as it is. You can write down a cow can be brown because you have seen a brown cow. You can write a cow can be black, a cow can be white. So you write down exactly what it is. And so you observe and you write down. You observe nothing more, nothing less. You write down nothing more, nothing less. And this becomes an essay on the cow. This is exactly the research process is. You do something, then you observe something. You observe a cell under a microscope, or you observed your Western blots. You observe the bands. So you observe what is there, and you write it down exactly what you saw, and that becomes your research paper. So observation is critical. If a cow has a black spot on its body, then you note it down that I saw a black spot in the cow. I don't know what it is. I have never seen it in other cows. If you see that, oh, I think I saw the toes of this cow were having some yellow color liquid, some yellow color liquid. That could be a pus. So that there could be an infection. and that is why there is that yellow color pus liquid on the toes of the cow. So you observe, okay, the cow is having an infection, and then you note it down, and then you go and give drugs. Well, when I gave this drug, the liquid disappeared, which means the infection subsided. Okay, this drug works. That is exactly the research is. Observation is critical. Now, when I was involved in the research work, which I just described in last 5 or 10 slides, we observed these bands here. Now, being a graduate student, you are not yet exposed to experiments. You do not understand what these square things are, what is the pink thing, what is all. I will just give an idea that a, the cells were lysed. We made juice out of the cells, like what, banana shake kind of thing. So we made a shake of the cells and we loaded it onto the gel, and this is what is here. The pink thing you see, the two lines, the two pink things, these are all the contents of the cells. So these are all the proteins. If you think if you see a thick band here, which means there is a lot of protein. These proteins are less here, some protein is less. So there are different sizes of protein, starting from 2 kd to 500 kd, and so 500 kd will be here somewhere on the top, and the smallest protein will be somewhere at the bottom. And this is all the gradient, all the size of the proteins, all the proteins coming from the cell is here in this one lane. Okay, now I observed that this there is one protein here which is more, and in this it is not. Now this was my sample, the wild type and the mutant. The mutant was showing more protein here, and the wild type was not showing anything here. So whatever is there in the mutant, it is not there in the wild type. This happened once. Then I did this experiment again, and I again saw that there is this protein, there it is more in the mutant and less in the wild type. This orange band, you see, it is a 75 kilodalton marker, protein marker. So this is around 70. This is 75, this is around 70. In 3 or 4 experiments repeatedly, I observed that there is a 70 kDa protein, 70 kilodalton protein, which is more in the mutant and less in the wild type. Now, this was unexpected, but I observed that there is a protein which is approximately 70 kDa and which is more in the mutant and less in the wild type. This was the observation. Now, I can only guess that 70 kDa, well, it can be HSP70, it can be HSP70, heat shock protein 70. All the heat shock proteins are of various sizes. The most known or the most studied one is the HSP70 and HSC70, and the 70 here in its name denotes that it is a 70 kilodalton protein. This was exactly the size which I observed in my Western blots, and it was unexpected. Now HSC70, as your ma'am already described you, that it is involved in response to stress, a lot of stress, any kind of stress you face, and it is involved. And it is involved in, so I just made a diagram here that it is involved in apoptosis, it is involved in autophagy, it is involved in a lot of protein transport, protein folding, transcription, it is involved in inflammation, it is response during hypothermia or hyperthermia. Hyperthermia means heat shock. Now, hypothermia is the part which the research from our lab added to its functions. Okay, now its name comes from, its name is heat shock protein. It is because this protein was discovered in approximately, I think, around 1970, 52 years from now, 52 years before this day. And it was discovered in the process when people were investigating the response to heat stress. What happens to your cells, what happens to your body when there is excessive heat? And then people observe that when there is excessive heat, these proteins level go up. the levels of these proteins is increased inside the cell. So while trying to survive excessive heat, our body generates this protein more, makes this protein more. So this protein must be involved in some kind of protection against the heat. And that is why the name came heat shock protein. All right. In last 52 years, 50 years, half a century, there are more than 5 lakhs papers, research papers on this protein alone. And all those 5 lakh papers, they deal with the role of this protein into n number of processes, multiple processes, and all those processes are well studied now. And most of this work relates to the response to heat, excess heat or heat stress. Nobody ever thought that this protein could be involved in cold as well. So nobody has actually studied the cold part, whether this protein can also protect you against the cold, or this protein can be responsive to cold, or this protein can at least sense cold or whatever. Nothing ever has been studied around this heat shock proteins with respect to cold. Anyway, uh, the disease mutant which the clinicians reported, the H443P mutant, which I was working on during the early stage of my research career, those patients had a unique problem. Their problem was that at normal temperatures, the patients are absolutely fine. But they go crazy. They develop hyperinflammation, rashes on their skin, fever, everything. Uh, once they are exposed to cold. And so this mutation, H443P, as the doctors reported, the patients are normal at normal temperature, 37 or above, warm temperatures, but the patients develop skin rashes, fever, itching, a lot of other problems when they are exposed to cold. This mutation also interacts with something that is at 70 kD and interacts better with this. We confirm those interactions that what I'm looking at, what I observed is actually HSC 70, heat shock cognate 70. So we now knew that HSC 70 is a protein that interacts with HP mutant much better than its wild type. It has higher affinity for the mutant. And then uh, the wild type also interacts, but it interacts like a basal level. So in this experiment, you could see if, okay. So here you see that only the HP mutant interacts better with the HSC 70. The other two mutations or the wild type protein, they interact, but they interact much less compared to the HP mutant. So this is something important, that the HSC 70 protein, the heat shock cognate protein, interacts better with a mutant H443P and LRC4. And this mutant is known to have consequences when there is exposure to cold. Okay. Uh, Next, we tried the same interaction at 4 degrees centigrade. HSC 70 and NLRC4 H443P. And you see here in the right hand side plot, at 4 degree, there is no interaction. At 37, there is interaction. At 4 degree, there is no interaction. At 37, there is interaction, whether it is wild type or it is mutant. This is interesting. HSC 70 shows interaction at 37. HSC 70 does not show any interaction at 4 degree. HSC 70 is selectively interacting with mutant protein or the wild type only at 37 and not at all at 4 degree. Now, doctors have reported that the patients start showing symptom and at a cold temperature, and that cold necessary need not be 4 degree. It can be as high as 28. So if the patients are at 37, they are fine. But as soon as they go to a temperature like 28, which is not very cold actually, it is actually okay temperature. It is a good temperature. But if they go at 28, they start showing symptoms, mild symptoms. You reduce the temperature 26, 24, 22, 20, the symptoms aggravate, the symptoms are higher, the symptoms are severe. So 28 is the temperature that is the borderline where the patients start showing symptom. So we tested the interaction of HSC70 at 28, and we figured that at 28 itself HSC70 begins to lose the binding ability. Heat shock protein with more than 5 lakh papers, mostly known for its response to heat, we discover that it does not interact with its clients at 28, at a colder temperature. Now, heat shock response protein is having a function and is having a response to cold. That is, we were very happy. We were also very scared that we are finding something which nobody else has found. Whether they will accept it or not, it is like 200 research labs around the world and one lab in CCMB in India, and then you are saying that something opposite is also true. We are not saying that heat shock response is not there. We are saying that it is there, but we are seeing that even cold shock response is also there. So while in research, it is very important that you are truthful to your observations, to your experiments, and once you have found something, you have to be firm on it. You have to present it with full confidence and with you don't have to back down. Hey, majority doesn't say so, so we should not we should not report it. I think it is wrong. That's not how it works. Many a times during the COVID times, many doctors did a good job because they went outside the guidelines given by their seniors or given by the authorities, the ICMR or the FDA or whatever, WHO and etc. Many doctors treated their patients with Ayurvedic medicines. With hydroxychloroquine was once a favorite job of for the doctors. Many doctors just treated their patients with warm water and simple antibiotics, depending upon how they perceived the symptoms of the patients. This was it. You don't have to always stick to the convention. If you have found something and you believe on it, then you should stick to it. And that is what exactly we also did, that we continued with our research and we found that I will just explain the end result of this research. So here I will explain. We know that NLRC4, that is the immune receptor, it causes inflammation, that is activation of caspase 1, and it interacts with HSC70. Now this is when everything is normal in a normal person. NLRC4 causes inflammation, it interacts with HSC70 and this. Once there is mutation, H443P mutation, NLRC4 becomes constitutively active. That it will cause hyperinflammation. That is the destiny for it. But HSC70 engages with this mutant protein much more than it engages with the wild type. It engages and it prevents its hyperactivation. So even though there is mutation, there is normal caspase 8 activation, caspase 1 activation. And HSC70 is having a protective role here for the patient. Because even though there is a mutation, HSC70 binds and prevents its activation and further caspase 1 activation. But at subnormal temperature, HSC70 loses its binding capacity. It cannot interact with its protein, with any of its client at 4 degree, at 28 degree, at 26 degree, at 25 degree, at 20 degree, any of the lower temperature, lower than 28, it cannot bind to its clients. So HSC70 comes out, it cannot bind. HP mutant is now free, and now when it's free of HSC70, it can cause caspase-1 hyperactivation, and it causes the disease symptoms. The patients which had HP mutant, they were absolutely normal at 37 or warmer temperatures because HSC70 was protecting them. As soon as those patients went outside or were exposed to cold weather, the HSC70 got out of the complex. It cannot protect them anymore, and the patients developed the symptoms. This is what, this is how we explained the pathology of the disease. That and this is how HSC70 senses the cold, and that is why I put the topic cold sensing by heat shock proteins. We also now know that HSP70, the other, you can say the younger brother of this protein, HSC70. So there is a whole family of HSPs, heat shock protein 90, heat shock protein 70, heat shock cognate 70, HSP27. And whole family, we think that the whole family of these heat shock proteins, they are able to sense cold. And they respond to cold. They respond to cold in this manner that at warm temperature they engage with their clients, and at colder temperatures they just sit back. They don't do anything. They feel dead. They don't function anymore. And if they stop functioning, the things go haywire. The people develop disease symptoms. So herein we were proposing that heat shock protein responds, senses the cold, acts as a let's say thermometer, senses the cold, and then responds to the cold in a certain manner by stop working. And then there are certain disease symptoms that follow through. Now, this was one disease where people were better at warm temperatures and they developed symptoms at cold temperatures. This was these people are not these patients were not from India. These patients were from Italy. Some of them were from Canada. Some of them were from Germany, and some of them were reported in USA also. There are a lot of diseases, other set of diseases where cold creates the problem or cold aggravates the problem. For example, arthritis. In arthritis, the joint pain increases during early hours of the day. It also increases during late hours of the day when the temperature is relatively low. Joint pain is relatively less during the afternoons. Arthritis pain increases during winters. People suffer more in winters than during summer season. Not just arthritis. The sensation of cold causes problem to the teeth. There is some clinical term for it. People are very sensitive to cold water if they have some problem in their teeth. That sensation issue is caused by some neuronal activity where cold water or any cold, even if cold air goes into the mouth directly, the people start having a very disruptive sensation in their teeth. And then there are medicines that say that okay, we will suppress the neuronal activity. That some toothpaste is there. There are some other creams that we prescribe. Now, anything in the body that aggravates, any problem of the body that aggravates because of cold, we can now have a hypothesis that HSC70 or heat shock proteins were the ones that were protecting you when you were in the war. And during cold, HSC70 stopped protecting you, and that is why you have the problem now. Or that is why you are facing more pain. That is why you are facing cold sensation, or things like that. Now, to work on this hypothesis that what happens to arthritis patients when they are exposed to cold, and whether there is any role of HSPs in that the problem of arthritis or the tooth hypersensitivity to cold water or things like that. Uh, so these are the some of the proteins that also respond to cold. These mutations in these proteins, which I have highlighted, um, they cause, they are absolutely normal. People are absolutely normal at room temperature or normal temperature, but the patients start experiencing symptoms when they are exposed to cold. So these are some other examples apart from arthritis and tooth pain, which I have just listed here. Uh, and where we think that heat shock proteins can be involved, that they function at normal and they stop working at cold temperature. And these are the hypotheses which you don't need to pay attention right now. uh, if you want, you can, this most of this is published. You can look for my name on Google Scholar, and you will find these papers, and you will find these diagrams there also. So anyway, to work on the arthritis part, and whether HSC or heat shock proteins are involved in the arthritis pain, which increases during winter or during early hours, I joined the division of rheumatology at this Harvard Medical School. And now here I am working on the arthritis part. Maybe sometime uh, we will, now this work here, what I am doing is now going to relate also to the odontitis. That is probably the gum bleeding and the swelling of the gum and the cold sensation. Because uh, whatever cells are responsible for the bone loss, the bone erosion in arthritis, the same set of cells are also involved in the bone erosion of, the erosion of the tooth, the base of the teeth. So maybe someday we will have some answers. My personal interest is heat shock proteins, but you never know. Like here, I discovered another protein, WDFY4, that have a similar role. So you never know how the research, which direction the research will take you. And this is the finding of what I have worked here, which I'm not going to explain, also because of the time constraint, and also a lot of information has been imparted to you in last 1 hour, so your mind must be uh, full of everything. You don't want to hear any more uh, of the science, most likely. But I will take you to another part, which is my own personal agenda, that I want to drive you people towards research. Apart from the clinicians, I'm not asking you to stop the clinical activity or stop being a doctor, but I feel that if even if 5% of you can contribute to research as a part-time job, it will be a great help for the research community. It will be a great help for your own patients, because the insights you will gain in a research lab will definitely help you to serve your patients better. It's not that you will write a different medicine, but it will it will help you to advise them better, actually. And patients need more of advice than a prescription from a doctor. Uh, this is my understanding. Uh, uh, as far as the research rewards or the research is concerned, um, there are a lot of things that you can add. Uh, of course, the satisfaction is one part. Of course, the benefit of the research community is one part. And I will just like to highlight the importance of the research part. During the COVID times, uh, the COVID testing became a huge problem in our country. There were lakhs of samples in a day, and how will you do the testing? The fun part is the junior most person in a research laboratory that we call as project assistants. They are not going to even get a PhD degree out of what they do in the lab. But the junior most person in a research laboratory, that is the project assistant, that is just MSc pass out master's degree holders who come to the lab and they are just trying to learn some research methodology. And these people were very much overqualified actually for the job of the testing. So the person who is considered of the at the basic pedal, the lowest pedal of a research wheel, research hierarchy, the project assistant, are the best qualified people who can do the COVID testing. Everything that COVID testing involves: designing of primer, isolation of RNA, PCR reactions, running a gel. This is daily routine in a research lab. Daily. You can blindfold me and I can do it, some of these things. How to tweak these protocols to get more effective data in lesser and lesser time. From CCMB, there is a guy called Santosh Kuncha. He just shortened the protocol for RNA isolation from 4 hours to half an hour. And it was just like that. And he got Young Scientist Medal, in which I was the nominee. I was also a nominee. So we were competing, and he got this award and not me, because he contributed significantly to the society in those times. You imagine, you give a sample today morning, and it comes back, the result comes back to you in the evening. And after his contribution, his discovery that, okay, let's do it. Let's do not do it this 4 hour protocol. Let's do it in half an hour. You could get your result in at 2 o'clock. If you give your sample at 7, you will get back a result at 2 o'clock. That was his discovery. And it was very simple. Everybody in CCMB could do that. So what I'm trying to highlight is, being in research sometimes, and especially in the times of things like pandemic, or many other moments will come where you are in a better position to serve the society. And of course, you can also earn money and recognition out of it. So yeah, try, if you find some interest, if you find a little bit of interest, try to join some lab for a couple of months, for 6 months, and test yourself if you are actually interested in it. You don't have to leave your clinical jobs. You should attend your clinic, you continue doing that, but also give some time to the research part. This is probably my request to all of you. And I hope I was trying to uh, impress upon you that how the research process works, rather than giving you uh, rather than giving you the idea of what I did into my research. But I was actually trying to impress upon you that how the research process works, how can it be more interesting, or it can be very interesting, and it can be very satisfying and rewarding. So I think I will stop here. If you have any questions, I hope you have, then you please ask.
Thank you, sir. Thank you for the wonderful lecture. You have explained in detail about heat shock proteins, how you had a different result from others, how you worked out, and all those. And finally, you have really motivated our students towards research, sir. Other than clinics, you have encouraged them to take part in research activities also. I hope our students are getting benefited through your advice. Coming to questions, we have two questions, as such. Uh, our participants would like to know if there is any role of food and drugs in HSP stimulation. If it is stimulated, will it have a beneficial role or harmful role?
Food and drugs.
Yeah, yes, sir.
Okay. So there have been actually many drugs that stimulate HSPs.
Yes.
One drug was invented, it is called geldenomycin. Now there are many derivatives uh, of geldenomycin. Once geldenomycin was in market for treatment of cancer.
Yes.
And uh, people thought that geldenomycin actually induces HSP levels, HSP 70, HSP 70 levels. And people thought that HSP 70 will uh, have some effect on cancer cells and it will kill it. Because in those days, around 1996, 97, somebody has discovered that HSP 70 contributes to apoptosis, killing of the cells. And so then and there, pharma companies jumped into finding that how to improve HSP 70 levels in the body. They discovered geldenomycin, and it works very well. It improves the HSP 70 level uh, to a great extent in your cells, in your body. And then they send it to the market. Doctors happily accepted it because doctors need drugs to prescribe to save their patient's life. Now, geldenomycin worked well. It improved the HSP 70 level, but it actually, and it also improved the symptoms in the cancer patients for first few months. And then the cancer will relapse and the patient would die like in a week's time. Uh, the thing is, now here I'm going to, that is why I also want, and this is one of the first instances when I thought that actually doctors should be part of the research process, because this drug was sent into market in around 1998 and was people doctors stopped prescribing it in around 2008 or 2010. The patients showed improvement for first few months and they died. Doctors knew it. Pharma companies did not. Every doctor knew that my patient died. Problem is, doctor doesn't know why. He just only knows that my patient is dying. He improves and then he suddenly dies. There can be n number of reasons that doctor cannot think of because he is not part of the research process. He was never exposed to how geldenomycin works. He only was told that geldenomycin works. Patient kept dying for 10 years. Problem is, HSP 70 level improves and it kills the cells, apoptosis. That was the research, that was true. But HSP 70 does a lot of other functions. It also helps the cells to survive. And it is more effective while it is helping the patient cells to survive. Most of its function or more of its functions are to help the patients to survive rather than to kill. Well, killing is one of its functions. That happens under a certain context when cell are under a certain kind of stress, when the cells are under a certain kind of signaling is happening. So under specific conditions, HSP 70 kills, but under n number of conditions, HSP 70 helps to survive. Now, well, the patients kept receiving geldenomycin. HSP 70 was up. It initially it killed the patients because there were more cancer cells in the body. But then cancer cells adopt very quickly, and the HSP 70 started helping them to survive. And the cancer cells survived, started surviving them. And then the patients would die in a matter of weeks. Have the doctors been part of research process as well? Or there will be some communication between the research and the researchers and the doctors. This thing will get highlighted that my patients are dying. What the medicine are you going? In next, in first 5 years, 2 years, 3 years, 4 years time, and then we might have stopped geldenomycin. So that was one story which I so food or drugs, they can induce HSP70, but just start using it is not a good idea because we don't know everything what HSP70 does. Like after 50 years of research, until 2 years back, we didn't know that HSP70 also senses cold. And so that is what it is. Uh, and the second part of the question was if HSP70 helps to survive. Yes, it helps to survive in more ways than we understand. Most of the cases, it helps us to survive or improve our health. But of course, there will be specific conditions and where it will also create problems for you. Uh, at this stage, I will also highlight this. Although I am a molecular biologist, I am uh, by definition a modern day scientist or a modern day researcher. And I am by definition a person who relates to the allopathy way of medicine. But now that I have spent a lot of time in this process, this research, and the allopathy is the direct outcome of our research, I also believe that the time tested methods of medicines should not be ignored. And that will create controversy, but it is it is also important. Maybe we should be the first persons who should go to the traditional methods of treatment or traditional methods of keeping your health, and uh, get some cues from there, prove it scientifically according to the modern requirements, because today unless you show the molecule, nobody will believe that it is right. That is why people don't believe in homeopathy system or in a Unani system or in Ayurvedic system, because you don't have a cell paper, a paper in nature, in paper in science for Ayurvedic medicines or homeopathic medicines. People just outrightly shrug it off. You will be like humiliated if you propose someday that homeopathy medicines also work, because there is no scientific data according to the modern research. So but of course there should be some way there, because the allopathy system and the research system has a lot of gap, and that gap is largely because of doctors not being in research and researchers not being into clinics. So either you fill this gap or look for other ways. Anyway, what is the other question?
Other questions, sir. Um, Actually, lichen planus is an autoimmune disease, no, sir.
Mhm.
It is due to high stress level. Will it follow the same pathway as it is in cold sensing, sir?
Autoimmune disease. What was the disease?
Lichen planus.
Okay, all right. Uh, Scientifically, I cannot answer this because I don't know this. Uh, if you are asking whether HSP will be involved or not in that kind of autoimmune disease or a stress-related disease or any kind of other stress-related disease, uh, I am not sure that it happens or not.
Okay.
But uh, my insight into it is, if you force me to say something, and if if I have to say something, then I would say that if not HSP70, there will be some other 70, or there will be some other protein which will be obviously involved.
Yes.
Uh, think about it like this: when you have an injury, if you cut your somehow you cut your while shaving, you cut here something, or you get some injury using a blade or a knife, you know that there is some injury in this part of your body. You can see it, you can feel it. So every time your hands go there and try to save it, or try to do something to help it heal. You apply some ointment and this and that. So you can see it and you do something to heal it. That's what I'm trying to say. Now, every now and then, your body is exposed to some mechanical stress, like you fall on the floor, you have some injury on your knee, you attend to it, you heal it, you apply some ointments. Now, during that fall, your knee, the skin of the knee was not the only thing that got injured. There were hundreds of cells that died inside your body in and around that area. There were hundreds of blood vessels that got ruptured here and there. Capillaries got ruptured. A lot of things has happened inside your body. Now you cannot see it and you are not attending to it. But there is something that is attending to those damaged cells, damaged blood vessels, damaged capillaries. A lot of things have got damaged. Now, who saw them? What happened inside your body? You never know. You don't see through your eyes to those problems. Now, your brain works in a certain manner. It has a certain mechanism that senses any kind of a stress, even mechanical stress. There are proteins that sense the exact mechanical pull. It has nothing to do with physiology. The mechanical pull of this there are proteins that senses these mechanical pulls. Like in physics, the exact things you read in the physics in class 12 is happening inside a cell inside your body. Look at some YouTube videos of actin proteins walking on the rail inside the cell. It actually walks like you walk. The actin protein, it holds a cargo on its head. Like people uh, have a box on their head and they are walking. You will see the actin actually walks like that inside the cell on a road. It's like a ropeway. Okay. Anyway, so I'm trying to suggest is any kind of a stress inside your body, whether it is in a brain cell or in any other part of the body, there is a mechanism that senses the distress and responds to it. So if not HSP70, there will be something that responds to it. And uh, uh, I don't know how to improve that. I'm not a doctor. I'm not any kind of practitioner or something like that. I can only do research. I can only ask questions and devise strategies to answer them. You have given me a question. So someday I will try to do it. Thank you for the question, by the way.
Okay. Our next question is there a link between gut flora and heat shock protein, sir,
Gut flora?
and HSP?
Yes, of course, there is. HSP70 particularly, not HSP70, sorry, HSC70 particularly, is expressed a lot more in the um, intestinal epithelial cells compared to any other cell type. It is actually almost 2% of the total protein inside those cells. So there is definitely some correlation, but I'm not sure what. We still don't know that particular thing.
Okay. So will that mechanism be same for this periodontal inflammation pathway also, sir?
I
HSP
am most
pathway?
definitely sure. We have not proven it yet, but I am more than 90% sure that periodontal pathway, HSP, will have a role in the neuronal cells. And maybe like if I continue to work like this, maybe in next 3-4 years, I can have some results for you.
Okay. Thank you, sir. Thank
Yes,
you for patient answering of our questions. Uh, to Ramya again, sir.
yes, sure. Thank you.
Thank you, sir. Thank you very much for accepting our invite and giving us a wonderful presentation on the pathway of NLRC4 and upper process, and also the research processes, and giving us valuable advice on research career. Thank you so much, sir.
Thank you.
I would also like to thank the institution, Sri Balaji Dental College and Hospital, Dr. Sumati Jones, Head of the Department of Pharmacology, for conducting series of webinar on DENTOPHARM 2022, Principal Dr. Jimson Sir and the Organizing Committee Dr. Satish and N.P. Murli. A special thanks to Dr. Nishat Sheikh and Angelin from Doc Mode Organization for the technical support. And I also thank the audience for their kind cooperation. Thank you.
Thank you.
Thank you, sir. Thank you so much.